Hello boys, welcome back to another interesting session of video tutorials, and this session is meant to class 12th biology students, and this is meant for 18th August 2020. Our memory always helps us in reflecting upon what we have been studying, and I'm sure all those who have seen yesterday's video must have been very happy about the experiment that Griffith has conducted. And all these experiments not only help you in understanding the properties of DNA and the secrets of our life, but also makes you a career. As I said, you can always become a molecular biologist in future. So this makes you to think. The very purpose of science is to make the student think rationally and logically, and I'm sure all these experiments will make you to think logically and rationally. So with that in our mind, let's move on to the next experiment. Biochemical characterization of transforming principle. What is that transforming principle that has taken place in Griffith's experiment? We were studying that there were two strains of bacteria, smooth strain and rough strain, S strain and R strain, and we understood that the mouse died when there is a transforming principle that has made the R strain to get converted. So what is that exactly, this transforming principle? They wanted to know. So we are thankful to three scientists. It's famously known as Avery, MacLeod and McCarthy. In 1933 to 44, look at that, 33 to 44, 11 years they have done a long experiment, and how much patience they should have. So thanks to Avery, MacLeod and McCarthy, they worked to determine the biochemical nature of the transforming principle of Griffith's experiment. And they have done a wonderful experiment. What is that they have done? They purified biomolecules, proteins, DNA and RNA from the heat-killed S strain. They added digestive enzyme of each, like proteases, so that proteases will digest the proteins, DNAs will digest the DNA, RNAs will digest the RNA. Like that, they added digestive enzymes of each to see which one could transform live R cells to S cells, smooth cells. So here, the purpose is they want to determine out of the three which is exactly the transforming principle. And what is that wonderful experiment they have done? Heat killed a strain they have taken, and to that they mixed an enzyme called protease. And when they mixed this protease and they added live R strain, and they could understand that transformation was taking place. Transforming principle was taking place. That means even though protease is added, the moment protease is added, what happens in this experiment? All proteins will be digested. In spite of proteins getting digested, still there was transforming principle taking place from R strain to S strain. That means proteins are not the reasons. Then heat killed S strain, smooth strain plus RNase. This time he has taken RNase. RNase is enzyme that dissolves RNA, plus live R strain he has taken, and still he was taking R strain. Still the R strain is transforming to S strain. That means in spite of RNA getting digested, the transforming principle was taking place. That means RNA is not the genetic material that is transforming. That's how they concluded. And very interesting one, heat killed S strain, that is smooth strain. Now this time they have taken DNase, an enzyme. DNase means an enzyme that dissolves or digests DNA, and they have taken live R strain, rough strain. And now very interesting observation they made, unable to transform R strain to S strain. So that's how they have come to a conclusion that it is not proteins, it is not RNA, but it is DNA which is the transforming principle. So you can see this in the test tube. They have taken culture medium, they have put that rough bacteria R strain and carbohydrates of S type bacteria, you can see, and then they have taken protein of S type bacteria, proteases, and the protease has dissolved the proteins, but still transforming is taking place. Then they have taken DNA of S type bacteria into smooth bacteria, and the DNA of S type bacteria with deoxyribonuclease, that is DNA. Now, when this has happened, there was no transforming principle. So, what is that they have concluded? Protein of heat killed S strain is not the genetic material, because when they put protease, the protease dissolved protein, but still the R is becoming S. And then RNA of heat killed S strain is not the genetic material. DNA of heat killed S strain is the genetic material, because when they put DNAs, because of that DNA got digested, and once DNA got digested, there was no transforming principle taking place. Hence they concluded that it is DNA, because DNA digested with the DNAs mixed with R strain, unable to transform R strain to S strain. But the biologists are not convinced, and at that time one more scientist in 1952, Martha Chase and Harsha experiment, they have done this another wonderful experiment which proves that DNA is the only genetic material, and this is proved beyond doubt by Harsha and Martha Chase. They worked on the virus that infects bacteria called bacteriophage. During infection, the bacteriophage first attaches the bacterial cell wall, as you are seeing here. It inserts its genetic material into the bacterial cell, as you are seeing here. You have got a satellite picture, that's bacteriophage, and all of you know bacteriophage is a virus, and the virus particle is entering into the bacteria. As you are seeing here, from A to B, it is just like there are receptor proteins in the bacteria which receive that bacteriophage, and in C diagram you can see the bacteriophage is replicating. In D, the numbers are increasing, and in E it's breaking. The bacteria is broken by the bacteriophages. So the viral genetic material became part of the bacterial genome. It manufactures more virus particles using host content. This is how even COVID-19 is also doing same.